Okay. So first I want to talk about the bioinformatics, because the definition of the bioinformatics is that it's a discipline that is concerned with acquisition, storage, analysis, and interpretation of biological data. And when people are talking about bioinformatics, most commonly it will be referring to the omics studies. However, bioinformatics is a very, very versatile field, and it's actually covering many, many subfields. So that's why when I was thinking what to present today, I actually decided that instead of presenting you the projects I have been working on recently, it might be of interest, especially in this forum, to show you the richness of the fields encompassed by the bioinformatics. And therefore, I decided to talk about my scientific road, where it started and where it actually led to. So the transitions between the different stages, they might seem surprising, but they had something very big in common, and it was the analysis of biological data. Okay, so this is what I spent my master's and PhD on. If you had more time, I would have asked you to guess what it is, but since we don't really have much time, I'll tell you what it is. That's a path traversed by a mouse within a 30-minute session when it was released in the circular open field, which is a behavioral maze. So what we have here is the animal that was released into the field, and then the coordinates of the movement of that animal were tracked across 30 minutes here, and sometimes the sessions are different. So you can always ask, why would anyone want to study such a phenomenon? Because it just looks okay, so some mouse is running somewhere. And my answer would be that locomotor behavior, and specifically its organization in space and time, reflects many complex processes, such as memory, learning, exploration, psychological states, such as anxiety, for example, and many, many others. So the structure of the paths, the velocities at the beginning of the session versus the velocities at the end of the session, they can tell us a lot about how the animal is learning about the new environment, because the environment here is novel to it. Also, if you see a change in locomotor behavior as a function of the number of visits to a location, then it can be an indication of a memory, because it means that the animal actually remembers that place. It remembers that it visited that place. So the behavior now is changing, not in time, but as a function of the number of visits. Also, if the animal is not moving, so then usually it's considered a sign of anxiety, and especially if that situation is changed, if we administer some anti-anxiety medications. So here, as I said, we see a path of the mouse that was released for 30 minutes into a circular environment. And we are recording the coordinates of the animals with a really high resolution. So we had 25 frames per second for 30 minutes, which ended up with 45,000 data points in time. So once you have so much information, you can do a lot in it. You can learn the patterns, you can look at the velocities, you can discover the accelerations, the stops of the animals, you can check the angles between different movement segments, you can look at the curvature of a path. There are many, many, many things that you can actually look at. And what attracted me to this field was that you can measure something so specific as coordinates and velocities, and that can reveal you the patterns in behavior, which seemingly looks very, very chaotic. So for me, searching for the patterns in something so chaotic was very, very interesting. That's why I started with this. And I actually spent quite a few years in it. So now I'll show you some examples of what I did. So when we released the animals, the mice in this case, into the arena, what we could see, and that's this plot that you can see here, that's exactly the same plot, but it's just longer. It's this one is 1 hour of exploration. So we could see that there are some spots that are visited many times by the animal, and also the curvature of the paths is much, much higher than anywhere else. And this phenomenon was corresponding to a very well-known term in exploration, which is called home base, which is a reference point that animal uses to learn the environment. So every time the animal will exit that point and will return, and that's how it builds the map of the environment in the hippocampus. So we really wanted to study it because up till now, the home base phenomenon was described qualitatively. So we wanted to study it in more algorithmical terms. So the first point was to actually be able to find it. And that's why I developed this algorithm that translated this map into a heat map, which was showing us the number of visits per location. As you can see, now suddenly all these 2 places that were of interest for us, they lit up. And the next step was to use image processing to find and really define these spaces. Once I could define them, then that's what I had. And then I could look at all the paths around them, all the organization of the behavior around these spots. So here we have 2 different examples. These are 2 different mice. Each one had their own spot. And then I could look at the paths going in and out. So for example, here we have a path going in, and then the animal exits here. Here the animal exits and enters from the same area of the spot. So we could look at the curvature inside of this place of attraction versus the curvature of the outside. We could look at the velocities because if the animal is running faster towards the space. So that would mean that it still is not as attracted by the outside as it is still attracted by the home base, and the home base is a secure space. It's a space where the animal would prefer to be at the beginning. So we could really learn a lot of a lot about the behavior organized around these spots once we could really define them. So the majority of my work during the master's was focusing on intact animals. However, during my PhD, I started working on the animal model of OCD. It's a pharmacological model in rats. And we examined these phenomena and some additional ones as well. Since I was working with the pharmacology and I was characterizing the effects of pharmacology, actually it led me to a very different field for my postdoc, because I got accepted to a postdoc to study the effects of pharmacological agents in breast cancer. So suddenly it wasn't mice running in the arena anymore. It was something completely different. But on the other hand, it's not really different because it's still analysis of biological data. So I was accepted for a postdoc by Dr. John Hassel in the biochemistry here at Mac, and that's what I was doing for my postdoc. So I just wanted to show you one of the examples of the projects that I did. I hope I still have time. So here I'm going to talk about the breast cancer and the gene expression profiles. In breast cancer, there are several molecular subtypes, and they're very important because they dictate the treatment the patients will receive. So it's very important to be able to determine the type of the tumor the patient has, because that's how the clinicians will decide on the treatment regimen. There are 7 known subtypes up to date. Some of them have relatively good prognosis, some have relatively bad prognosis. They're all different. And there is one subtype which is really rare. And its problem is that it hasn't been defined in a clinic. So it means that you don't have IHC markers to use in the clinic to be able to define patients with this type of cancer. And the main problem is that this type of cancer has a very poor prognosis. Up till now, it has been defined with the transcriptomic studies, but nobody could define it with in the clinic with the IHC staining. So the goal of this project was to discover the biomarkers that will help us to define this subtype in the clinic. In order to do that, I used this large collection of 1,600 tumors. I compiled this collection using 7 or 8 independent breast cancer datasets. And once I compiled all these samples together with all the batch effect removal and all the appropriate procedures, then I use the classifiers to perform the subtyping, the molecular subtyping. And that's what we see here in the barcode. So different colors refer to different subtypes. I will not talk about the specific subtypes here, but just to show you how rare the subtype we're talking about is. And this type is called the Claudin-Low. So the Claudin-Low subtype, out of 1,600 tumors, we had only 80 samples being Claudin-Low subtype. And they're marked in black. As you can see, you barely see any black lines here. So the first step for me was to compare the Claudin-Low subtype to all the other subtypes in a pairwise manner. And then I found the 130 genes that are differentially expressed between the Claudin-Low subtype and all the other subtypes. And that's what I'm showing here. Now we have the samples of the rows, so you can see the different subtypes. Black is our Claudin-Low subtype, and this is the signature that I found. So the next step was to validate that signature. The main problem is that you either have the IHC or you have the transcriptomic profiles. You usually don't have both. That's why we went to a substitute to the human cell lines, because the human cell lines Some of them are known to be Claudin low, and we can always do the stainings if you want to, and we can always get the transcriptomic profiles for these cell lines. That's what I did. I took the signature of the 130 genes, and I checked whether it's characteristic to the Claudin low subtype in cell lines versus the other types of the cell lines. And it actually was. So it was a very good validation to a certain level of what we found. As a next step, we also, let me see, so then what happened was we couldn't really proceed with 130 genes because there were too many genes, so we had to reduce them. So I selected the top differentially expressed genes, and we added a few candidates from the literature, so we ended up having 11 genes as markers, as possible markers. So what we did, we used the TMAs of these human cell lines first to stain the cell lines with these markers to see whether by using these markers we can actually identify these the Claudin low cell lines, and we could. So that was an additional validation in the cell lines. Next, we moved to the human tumors. With the human tumors, we had a very large cohort of 942 tumors, so we had them in TMAs. However, the problem was that we didn't really have information about any of them being Claudin low because we only had the staining information for them, so we didn't have the transcript for them. That's why all we could do here, unfortunately, was to see the prevalence of the tumors that we defined as Claudin low, and the prevalence was very similar to what you would see in the general population, and also the survival rate was also very similar to what you would see. So in general, we said that given the limitations of the study that we had, we did find a few markers that could be used in the clinic to predict the Claudin low subtype. See, so now I would just like to thank everybody I worked with. So it's the doctors Ilan Galani, Henry Schachtman, John Hassel, Anita Bain, and other collaborators I'm working with. Thank you very much.
Awesome. So if anybody does have any questions, feel free to raise your hand or type it in the chat. But I guess we'll get started with a question just so we can get the ball rolling. So one question we had was, what developments do you think have to take place in bioinformatics to transfer your current knowledge towards a clinical context as opposed to a laboratory context beyond breast cancer?
So what you're asking is, how can I translate what I'm doing in the research to the clinic, or is it a more specific question about the breast cancer?
How can you expand it beyond breast cancer?
In order to expand it beyond the breast cancer, well, I will not say all I have to do, but the main part would be to obtain data sets that are related to the disease in question. And that's what I'm usually doing. I'm compiling large data sets using smaller independent data sets. And then we study the disease in question. So first you have to find the disease you would like to study. Then you find the data sets and you start working with them. And usually if you have many data sets, you would try to compile them into larger collections. So you train, you validate. Once you get some results, then you want to validate them in a more clinical setting if possible.
Awesome, thank you. So if anybody wants to raise their hand to ask a question, you're free to do so. Fatima?
Yeah, hi. So thanks so much for your presentation. I just had like a little specific question about the first case you were talking about with the mice and in the circular environment. So you said that there was like a reference point basically where the mouse would most likely like come back to kind of examine the entire surroundings and see what's there and kind of explore and map out the area. So I don't know if you've had like other similar experiments, like, do you know if this reference point is just random or if there's like some type of science behind why they chose that type of spot?
So usually with the mice, it's relatively simple. It would be the spot where they're introduced to the arena. It's either that or it will be near the wall, because mice and most of the rodents, they prefer walls. So they would not enter into the center unless there is a reason to do so.
Okay, thanks.